51 years ago when a group of neurologists realised the importance of establishing an organisation to fund research into neurological diseases and disorders. 1 in 5 New Zealanders are affected by a neurological condition, and there are over 700 neurological conditions. We receive absolutely no government funding, so we are completely reliant on the generosity of New Zealanders supporting us. In the past year, the Foundation has funded close to $5 million of neurological research, and because of the generosity of our supporters, that's nearly $1 million more than the previous year, which is really, really good. Also, during the COVID pandemic, the Foundation committed to supporting the research community by granting time and financial extensions. We were the first and, for a significant time, the only organisation supporting researchers in this way. It's absolutely vital that research continues in order to one day find preventions and cures for these really awful diseases. So as well as funding research, the Foundation has made a commitment of funding the Neurological Foundation Human Brain Bank for another 5 years. This resource is one of only 8 in the world, and it's a world-class resource which is headed up by Professor Sir Richard Fall. The Foundation has also committed to funding the Chair of Clinical Neurology for another 5 years. The focus of this, since 2008, has been on stroke and the clot retrieval procedure. The Chair of Clinical Neurology is headed up by Professor Alan Barber. At our most recent funding, at the beginning of June, the funding round, $2.2 million worth of neurological research has been funded, and our next funding round comes round in November of this year. We are so incredibly grateful for every single dollar that we receive in donations. The research cannot happen without the support of our donors, so thank you very, very much. It now gives me great pleasure to introduce tonight's speaker, Professor Anne La Flamme. Anne is a Professor of Immunology at the School of Biological Sciences. She has a primary research in the immune regulation of disease. Recently, she's been working on identifying new therapeutic targets and drugs to treat multiple sclerosis. Anne has been collaborating with national and international researchers to develop therapies to promote repair of the neurological damage that occurs during multiple sclerosis. Over the past few years, Anne has been the recipient of a number of travel and research grants from the Neurological Foundation to gain a greater understanding of MS and to discover new treatments that will benefit all people with MS. So after Anne's talk, there will be an opportunity to ask questions, but in the meantime, welcome Anne.
Move forward. Good. That works. Thank you very much for the invitation to talk and really to present in a slightly different way, because usually I talk about mechanisms and, this cell does that and really diving in there. This is slightly different because I thought, really, let's think about how we get from the idea, that research idea that you think is this beautiful discovery, how do we bring that to reality? And in this case, because my specialty is in that drug development, developing therapeutics, how do we do that? And so I'm going to tell a few tales that build upon that idea. But first, you really do have to know where I'm coming from so you understand my perspective. So I was born in Maine, the southern part of Maine, now living in the southern part of the North Island. So I think I really like these small little areas. I don't think I can get this. So I can't get the pointer, so we're not going to do the pointer. But that's where I'm from, and now here in Wellington. And it's important to know what I am is an academic. That is who I love to teach, and so that's why I'm here. I also run the MS research program for the Malaghan Institute, but I'm based at Victoria University of Wellington, probably teaching one of your children, your grandchildren, or one of your neighbors, as well as postgraduate students that are doing research. And they are all part of this. And so, although I'm talking about it, this is not going forward. Let me see. Excuse me? Ah, there we go. No, it could have been me. Not doing the pointer. Okay. So, as I said, it's not just me that has done all of this work. So there's a huge team of students and researchers that have contributed to everything that I'm going to talk about. Even though I'm here, really, all the glory, if there is any, goes to them. Okay, so this is just a recent photo of the group, but this is over many, many years. I will talk about research. So one thing that has driven, or two things that have driven me, questions that have driven me in research are, what, I have to look at this. I usually get to see what my slides are, sorry. So, why is this happening, and what can I do about it? And really, that's at the base of the MS research program, which is understanding. Thank you. I think I'm going to stand a little bit to the side, and that might help a little bit. So again, the why this is happening, understanding the mechanisms, the why. I'm a scientist. I want to know why things happen. But I also then want to know what I can do about it. And my background is in developing therapeutics. That's where my strength is, and so that's what I say, that's what I'm going to do. And when you look, sorry. Okay. Can I ask, are you doing them forward, or is this finally working? Okay. I'm going to just check. Do I have to point it elsewhere? This is not very... Sorry, excuse me, I'm going to stop. There we go. Well, let's get back onto this. So I'm going to talk about two different types of approaches for what am I going to do, or how can I help? And the first is thinking about repurposing drugs. So thinking about how you can take a drug that has been used for years and put it to use doing something different. So I will tell that story. I have no idea why this is not working. Okay. Should I just say next slide? Yeah. There we go. So, so then also I'll tell a story about thinking about new drugs as well and how those have developed. So the next slide, please. So when you think about drug discovery and you think about going from that idea, how do you do it? Well, you can sum it up into 5 steps, going from that discovery, the light bulb going off, maybe a few tests, to then starting a preclinical assay, which may be dealing with more specific cells or animal models, and then going into your clinical trials, where you start to really see whether or not what you have is going to be useful. Once you've gone through that clinical phase, you then need to get approval through a drug approval agency like MedSafe or the Food and Drug Administration in the US or the European Medicines Agency. So all of those need to review your package of data to give you approval. But it still goes on because there's what they call post-marketing surveys, where they're looking for safety once a drug is out in the population. Next slide, please. Now for me, or for us, we look at those 5 steps, but it's not one person doing all of those 5 steps or one organization. Currently, a lot of the first 3 steps, the discovery, the preclinical, and the clinical trials, the start of those, most of those are really housed in research institutions, and many of those are academic institutions. And being housed in academia makes sense because it is about that discovery. And because within that discovery, there's a lot of what you might say is failure to go on and progress to the next stage. But with each failure, you often learn so much more, and you learn a lot about the why when you find out how different drugs or compounds or therapeutics will work. And somewhere around that clinical trial stage is now where you have commercial organizations that will take over and oversee that direct development pathway. When you look at the ideas that you start out with and the end product, you start out with a lot of ideas. Often, I think they estimate for every 5 to 10, 20,000 compounds you may start with, you're going to start narrowing that down to maybe 250 going into the preclinical work, maybe only 5 going into a clinical trial, and only 1 finally making it out. So in some ways, it's a bit like a lottery, where you have a little bit of control, but at each stage, as you see those numbers being read out when Lotto's going, at any moment it can fall over. We do have a little bit more control than Lotto, which is good, and good ways to predict. But what I want to say is that there's a lot of investment in that beginning portion, but that's also why it's really good that we have the discovery aspect. We have the that we are learning more and more about what is happening. Can I have the next slide, please? So I'm really now just going to focus on the first 3 stages, because that's what I've been involved in, and that's what my vignettes are going to cover. So going from that idea to the preclinical, and then getting into that clinical trial. And I thought it's important really now to think about how we test things, how we know that they work in people. And to do that, the very first thing you have to do is that phase 1 clinical trial. And what that means is you want to make sure that it's safe. So they're often done in a very small number of people, in often healthy volunteers, just to ensure that they are safe and that you have an idea of the dose range. From there, you can go on to the phase 2 clinical trial, and that's where you're going into people that have that condition, so that you can again verify safety, dose, and start to see the signals that tell you that something will be efficacious, will work to treat that condition. And then it's that last stage, the phase 3, where you start really looking beyond that small population, you go into much larger populations of people with that condition to show not only that something works, but that it is better than what is out there. And better maybe because it works better, but it also may be better because it is more suitable, it is easier to administer, it is just safer as well. But that's really important at that phase 3. Could I have the next slide, please? Okay, so I'm going to tell you 2 vignettes. The first one is about looking at developing and repurposing atypical antipsychotic drugs for multiple sclerosis. And then the second one is about remyelinating therapies. So as you can see by the arrow, there's one that covers a much wider range. And so I'm going to focus mainly on that. So if I could have the next slide, please. But to tell you that, I have to just highlight a few points about multiple sclerosis and what's happening there so that you can understand why those drugs were chosen and what we are trying to do. If I could have the next slide, please. So for multiple sclerosis, it is about the myelin sheaths. So what are those? Well, when your nerves signal from one neuron to another, they signal along a nerve axon, and you have to send a signal. Just like an electrical wire, though, you need to insulate that. You need to control the signal strength, the speed, and make sure that it is effective and efficient. To do that, those axons are covered by myelin, and myelin provided by a specific cell type, the oligodendrocyte. And they wrap themselves around to insulate and form these layers. So if you look at the axon, you can see there's a myelinated axon, and you can see the green wraps of myelin. That's a very healthy situation. Now, what happens in multiple sclerosis is that myelin itself becomes the target of the immune system. So it is a disease that is driven by the immune system and by cells coming in and attacking that myelin, destroying it, and then leaving those neuronal axons exposed. And that interrupts the signaling of that nerve. And depending upon where those nerves are that have become demyelinated, you can experience the symptoms of MS. And that goes from the visual disturbances, cognitive disorders, impaired movement. There's a whole range of symptoms. But you think about what your brain controls everything. And so there's that potential for it to affect many, many different systems. So the outcome of that is that you are going to have damage, but that damage can be repaired. But if you have consistent attacks, you can then have relapses. Could I have the next slide, please? So that's really kind of shown in this slide, where if you compare the healthy situation, down below that you can see that in the form of multiple sclerosis known as relapsing remitting disease, you have that infiltration of immune cells into the central nervous system, causing damage, destroying the myelin sheaths, and leading to attacks, the disruption in the signal. That rapid damage can be resolved because the immune system does not like to be on. It likes to turn itself off. Okay? And when it turns itself off, there can be repair, slow repair to remyelinate axons. So relapsing remitting, having those distinct attacks, is the most common form of multiple sclerosis, and about 85% of people with MS will develop that first. Now, 50% of those, at some point, will have a shift in how their disease progresses and go from having distinct attacks to having a slow progression and progressive accumulation of disability. And that happens to about 50% of people that start with the relapsing remitting, and some people start that way. And with this, there are no longer, or it's believed there are no longer, these immune invasions into the central nervous system causing this damage. Instead, the immune response is compartmentalized, so it's within the central nervous system where it is causing slow but consistent damage. And when that slow, continuous damage occurs, there's also no repair. And so, in addition to having demyelinated axons, you often will have damage to the axons themselves, leading to axonal loss. And that can cause permanent disability. So when you look at this, you say, what can I do about this? Could I have the next slide, please? Well, a lot of people have looked at this and said, what can I do? And in fact, there's been a lot of success in treating the immune component. So all of the current MS therapies target those immune cells that invade into the central nervous system, that invade into the brain, into the spinal cord. And usually they work by targeting them outside of the brain, by turning them off, by removing them, or by preventing that invasion into the central nervous system. So that is really successful for the relapsing remitting form. But for the progressive stage, progressive MS, it does not help, because we're not dealing with immune cells that are outside of the central nervous system, but those that are inside. And so for that, we really need we need compounds or therapeutics that will target that chronic inflammation that is inside of the central nervous system. Okay. But we also need to think about how we can promote recovery. How can we help that remyelination? So not just prevent the damage by the immune cells that are from the outside of the central nervous system or those that are inside, but how can we recover? Next slide, please. So those are the two targets I'm going to talk about. And the first, thinking about trying to quell the inflammation that's inside of the brain, led me to this story, which is looking at the atypical antipsychotic drugs and trying to repurpose them for MS. Next slide, please. So this very much, the discovery phase, with repurposed agents, discovery phase is not necessarily about identifying the compound and doing necessarily biochemical assays. This one was actually at a Neurological Foundation event where I was talking to Bronwyn Connor, who is a neuroscientist, a fabulous neuroscientist up at the University of Auckland. And she started talking about risperidone, which is an atypical antipsychotic agent. And that sparked an idea. And you might be starting thinking, why would that ever spark an idea? Well, let's think about what risperidone does. So risperidone, when I say it's an atypical antipsychotic agent, what it means is it treats psychosis. Psychosis such as that you would find in schizophrenia. And it was developed ages ago. So you can see in 1988 it was developed, and it was developed so that it would target a particular neurotransmitter receptor, which is a dopamine receptor and a particular type of it. Now, all antipsychotic agents target that receptor. And the original ones, the typical ones, only targeted that receptor. But risperidone, when it was developed, it targeted a few other things. And so it was called atypical. Good, so I've explained the name. Why would I be interested in it? Well, I was interested in it because Bronwyn was talking about how it had anti-inflammatory activities. And I thought, there you go. something that will quell inflammation, and something we know gets in the brain. That's what we want. So we started looking into this and realized, we shouldn't just target one, we should look at another as well. And so we selected risperidone, which is one of the first of the atypical antipsychotic agents, which was developed back in the 50s. And this as well was shown to have anti-inflammatory activity. Okay, next slide, please. Oh, I should say that both of them also had been repurposed for other things as well. So their expanding uses were there. Could I have the next one, please? Okay. So that meant we could go directly from discovery into a preclinical model. And we use a preclinical model of multiple sclerosis. What that means is that we induce a progressive paralysis that we can measure. And as you'll see from this, the paralysis develops, and then it starts to resolve a bit. But it is a myelin-specific response, so we know that it is the immune system infiltrating into the central nervous system, targeting the myelin, and causing the damage. So it's myelin specificity, it has demyelination, and it has a relevant clinical outcome, which is the paralysis, impaired movement. So this model actually was developed back in the 1930s, so it's a very well-established model. Okay, the next one, please. So I'm going to start showing people's pictures just to help recognize there were a lot of people that have contributed to this. But the first thing we did was we tried out risperidone, we tried out clozapine, both of them worked, and then we compared them. And what you can see from this graph in the middle is if you look at the risperidone, which is in the little red diamonds, there was a significant reduction in disease when animals were treated with risperidone. And we treated by putting it in the water so that they would have it orally administered, and they would just drink the risperidone. Similarly with the clozapine, and with the clozapine, we found there was a dose-dependent decrease, and that higher dose of clozapine, which is in the blue, was very effective. We also found it was effective if you used it before disease, at the onset of disease, or even during chronic disease. We found that it was effective at reducing that paralysis. So together, we said, okay, this is just what we want, something that's orally administered. We found that both are effective, but one appears to be better than the other, and it has a relevant outcome. And it's effective after disease onset, which is really important, especially if you're targeting a progressive disease. And targeting MS in general, you're only treating after the disease has started. Next one, please. So, I'm a scientist, so I want to know why. How is this working? Okay, and it's always important to know how things are working. Does it work the way we think it's going to work? And what we know about how MS works and how the immune system works is that while the immune cells that target the myelin, that say this myelin needs to be destroyed, those are T cells. However, the cells that actually do the destruction are going to be other immune cells, and in particular, macrophages or monocytes, or in the brain, the microglia. All of those are very related. It's like 3 different shades of the same color, okay, for those cell types. So we first looked to see whether or not it was the T cells. Did the clozapine or the risperidone directly alter the T cell? And in fact, Peruz, who's the first smiley face on your left, he did a lot of work to identify that, in fact, it was not the T cells. It was not directly working on the T cells. We were working on another cell type altogether. And through quite a bit more work, we identified that if you looked within the central nervous system, if you look within the brain, and these are looking at tissue sections in the cerebellum, so part of your brain, and looking at the microglia, you can see that those cells, it looks like there are these purple stars. Those are the microglia that are very activated. And in MS, they become very activated. They're those dark stars. But when we treated with risperidone or clozapine, we would find that that was quelled. They were no longer, they were there, but they were no longer as activated and causing problems. And so we had identified that it was the innate immune cells, so the cells like those macrophages, the monocytes, that were being targeted by risperidone, by clozapine, such that they wouldn't drive this damaging response. And then we thought, okay, this was what we wanted to see. We wanted to see that these anti-inflammatory effects would do what they were supposed to do inside of the brain. But can we enhance repair? And so Nikki Templeton, who's the middle smiling face, did a project trying to understand whether or not it would enhance remyelination and repair. And using a model that did not rely upon the immune system causing damage, but being completely independent of the immune system, she was able to show that it would enhance repair. So here we had two drugs. We had clozapine, we had risperidone, had been used for decades. We knew a wealth of information about them. We knew they were anti-inflammatory and quelled that inflammation inside of the brain. They reduced disease, and they enhanced remyelination. And we thought, we're there. This is great. Could I have the next slide, please? So we thought, there we go. Let's think now about the next steps. Well, as it turns out, what often has to happen behind the scenes when you have a discovery, when you have something, you need to protect your idea. We are now in a world of protecting our ideas. And for these, you protect them through a patent. And often, the idea is you start with the idea, you get as much evidence as you can to support that idea, protect it, patent it. But that process is a little bit more obscure than you would imagine. And often it's a little more difficult. So you do start out with a provisional patent, which you will file, and that sets a date. It gives you a year to file a full patent, where you put all of the information you can to show that you have just the most amazing thing in the world. Well, in fact, what you have to show is you have two criteria that it becomes examined on. It's examined on novelty, and it's examined on inventiveness. So novelty is actually quite easy. Has anyone ever done it before? And usually you know that. You can search that out and find it. Has anyone ever reported it, done it before? The second one, though, often is a big stumbling block, and it's a big stumbling block for repurposing, which is what I found out in this. Because inventiveness is basically that somebody who was skilled in that area would not have come to the same conclusion as you, that you have gone beyond what was just a reasonable next step. And you might sit there and say, well, how do you do that then? You know, isn't it always the logical next step that's there? But that ends up being a difficulty for repurposing, simply because that product has been used for so many years. And people somewhere, at some point, may have said, oh, that could be used for this. Or they've done an assay and said, oh, now I can cure everything in the world with this. So it turns out it's very difficult to patent for repurposing, because it ends up being a method of use you do. However, we did succeed mostly. However, that was all going on in the background because, in fact, it's not often done by the scientists, but by lawyers. So that was the extent of my knowledge in this. Okay. Could I have the next one, please? Okay, so while that was all going on, I was centered on, does this work? You know. But before going to, does this work? When you're repurposing a medication, you know a lot about it. And I knew that there are side effects with risperidone and with clozapine. And so these were drugs that had been used for decades, but they had never been used in people with MS. And so instead of kind of jumping in and saying, let's have 100,000 people, the first step was the baby step, which was, is this a good idea? I'm a scientist. Is it a good idea? You always do little preliminary tests to make sure that you're on the right track. And so I ran a trial, and it was with the vast experience and help of the neurologists at Wellington Hospital, and Dr. David Abernathy was absolutely fantastic. He and David Burke were the neurologists that were primarily looking after the trial. So again, this is another one where I'm not a clinician, so I step aside, I let them do what needs to be done. But it was a randomized, placebo-controlled, and blinded trial with the idea that we just wanted to know whether it was safe. Maybe get hints of efficacy, that there was a signal, but it was about safety and acceptability. So we had primary endpoints of looking at that. We also knew that a lot of the side effects with risperidone and clozapine were dose-dependent. And so we targeted very low doses, and from our preclinical work, we knew that low doses were effective. And so this was just ensuring that even with those low doses, there would be safety. Could I have the next one, please? Okay, so we're designing a trial. And a trial for MS is sometimes very, very difficult depending upon what you're looking for and what you want. So in designing a trial, one of the key things you have to remember is that you have to pick the right primary outcome, meaning that's your measure of a success. What do you consider successful? That is all done before you even recruit the first patient, before you get ethics approval. Everything is done first. It often gets published or it's put on a registry because once your trial is designed, you don't get to shift things around. Okay? So before anything starts, you say, my trial will be successful if this happens. So you have to be really careful about what you pick. So we were choosing as a primary outcome for safety, we were looking at two things. The first was adverse events, and the other was a treatment satisfaction questionnaire, because we wanted to know whether people thought it was suitable. So then you have to think about your sample size, right? Because if you have a primary outcome, this is what you're measuring. You have to make sure you have enough people that you can find a difference, if there is a difference. And so that's often very hard. You can't have too few, because then you can't detect a difference, and you can't have too many, because one, logistically it's very difficult, but two, if you have too many, then you're exposing a lot more people to a potential risk. So you need to find just the right balance. So one of the things that complicates clinical trials for progressive MS is that we don't know what that outcome is. Because especially in progressive MS, how people present, the symptoms they have, the rate of progression is very, very different. And there isn't truly a consensus as to what is the best primary outcome measure. If you do look at progression as a very crude one, says, are people getting worse? you often need a very large number of people over a very large number of time, a large amount of time, and that makes it incredibly difficult. And it's one of the things that I think has impeded a lot of clinical trials in multiple sclerosis, is simply trying to pick out that outcome and a measurement that gives you an idea if something works. So we designed ours to have a block randomization. And this becomes important later if you're wondering why I'm saying this now. So we knew how many people we wanted to recruit, and they were going to be randomized. But randomization, having one group first and another group and then another group is just as random as having them completely mixed up. So we blocked them so that we knew that there would be at least an even spread throughout the course of the trial. And then we said we're going to have a safety review after our first block and just see what happens. Next slide. So we did that. We stopped after our first block, and we did our statistical analysis. That was all done by our statistician, and then we unblinded the groups. So we didn't know until that point what people had. And when we did that, we found, if you look at the graph in the middle, looking at the percent remaining on trial, this is just looking at people that have withdrawn from the trial. You can see the very top black line is the placebo. If you look at the red line, that's the risperidone, and the blue line was clozapine. And it was a very conclusive result, only from that first block. And what it showed us was that within the first week, people who were receiving clozapine developed, I guess, what was called a rapidly reversible deterioration, basically in lower limb strength. But it was rapidly reversible. People were withdrawn, and that the weakness in the lower legs went away. And the problem, one of the big problems we saw there was that it was causing people to fall, which is the last thing you want. So we looked at that, and I said, that's really quite strange. And that's what was happening in the clozapine group. With the risperidone, it was just not acceptable, it's not suitable when we went back and looked at those surveys. So you can see that for both of those, even though they were given at very low doses, they were not considered suitable or tolerable by the people that were taking them. And in one case, in the clozapine, they were certainly causing an effect that was unexpected. The other thing, while we saw an unexpected effect, we didn't see the expected effect. Because one thing that we knew that clozapine would do is that it causes, the first time it's given, the second time it's given, it causes a drop in blood pressure. And that's usually within the first 4 hours, and then it stabilizes, and that goes away. But we didn't see that. And so we were it was a trial of very unexpected findings. As a scientist, I was fascinated. I still am. Looking at developing something that is going to be useful to help someone with MS, it was devastating. Okay, so I think I'll just go to the next slide. So it left us with, does it actually work? Because what that showed us, these are our findings, what did we learn? Well, we learned that you can't just take a drug from one indication and put it to the other without being very cautious, because a chronic disease like MS can change the baseline, the system, can alter your physiology such that it may mean that a drug which would normally not be a problem is problematic. So our work suggested that there were changes actually to the pathways that were targeted by clozapine. So something had changed there. And so we've been following that up to try to understand what those are. And actually I've had a very talented postdoc, Katerina Robuchon, who's been following that along and really found some incredible discoveries about how those changes occurred. But it left me saying, would it work? I still want to know, would it work? If it were tolerable, is that a target that is a useful target? So I did look through data, and this is data that was collected by another research student of mine, Carl Bears. He did this a few years ago, where he was looking to see whether or not clozapine affected the immune response, immune cells from people with MS. And he took out the monocytes, so this cell from the blood, and he just cultured them, sat them there for, I think, 8 hours, or he cultured them with clozapine, just to see what would happen. And he compared monocytes from healthy people to those with MS. And when you look at healthy people, you can see they don't produce any of this particular molecule. And this is a molecule called interleukin-1 beta, which is associated with driving an inflammatory response. Those monocytes don't normally produce it. You give them clozapine and nothing happens. That's not much of a surprise. But if you look at what happens in the monocytes from people with MS, you can see that they normally produce this. They're normally driving that response. And if you add the clozapine, it's reducing it. And so that was a hint that, in fact, it's a good target. It's just not the right drug necessarily for it. So I also learned from this that when you have a good idea, somebody else probably also has that good idea. And about the time we had been running the trial and organizing for the trial, there was another group that started. It was a group based in Calgary looking at quetiapine, which is another atypical antipsychotic based upon the structure of clozapine, but it was found to have fewer side effects. But on the flip side, it's also less effective. Next slide, please. And following that up, you can see they were looking at the quetiapine for myelin repair, which fit in with the fact that clozapine was also enhancing remyelination, so it was all working really nicely. And a clinical trial was being run. And just when I was preparing for this talk, I noticed that there was an abstract from a talk talking about that trial. And you can see quetiapine is not tolerable to people with MS in doses potentially required to enhance myelin repair, which shows it's that whole class of agents that may not be appropriate in this case. And there's something about that pathway. Now I'm still really fascinated. There's got to be something in this pathway with that. Okay, could I have the next slide, please? So that was the full story of that, where we've gone from that idea, developed it, gone through the trial. We went through the patenting, but we fell over. We fell over in the five clinical trial, which is quite sad. But we have continued to look for other targets. And now we've been targeting not so much that inflammation, the chronic inflammation, but really trying to find those agents, those therapeutics that can remyelinate, that can help repair and recover. And part of that is because that is important in progressive MS, but it's also really key in relapsing remitting. So it is a commonality to all types of MS, is that demyelination. Could I have the next slide, please? Okay, so this work is done in collaboration with Bronwyn Kivel and Thomas Provenzano. So Bronwyn Kivel is also here at Victoria University of Wellington, but Tom is a medicinal chemist at the University of Kentucky. And those two have worked for ages on a molecule or a receptor known as the kappa opioid receptor. And it's a receptor that is really important in analgesic effects, so that's anti-pain and moderating pain, but also is associated with dysphoria and aversion, some negative effects as well. But they're through slightly different pathways. In any case, we know that that target, or they've been working on that target for years, trying to find non-addictive pain medications. Because you probably see the word opioid and think of the opioid crisis. But in fact, that all has to do with another opioid receptor. And so, in fact, this receptor does not tend to have kind of addictive properties and is a target, in fact, for anti-addictive treatments. In any case, they've been working on these for years. And suddenly we started thinking about, and again, it was a random conversation starting things off, and we started thinking about how to apply this to multiple sclerosis and how this would work. Could I have the next one, please? And so here we can start with our preclinical, and we started with our preclinical because we found a repurposed agent, which was called nalfurafine. In fact, it is the only clinically used kappa opioid receptor agonist. So we used that just to see if it would work. And we put it in our model, and we treated only at onset, and it worked brilliantly at completely reducing disease and enabling that recovery. So we used it, again, this is therapeutically. We've also used it well into the chronic stage of disease, and it worked well. And so this is work done by Lisa Denny and Katerina Robuchon doing this. So the next one, please. I'm going through these very fast now, so you don't have to worry. But again, the question was, will they enable remyelination? That was the big question. And will they address the other effects that you have in MS, such as that inflammation into the CNS? So if you look at the graph, which is off to the left, we looked at whether or not immune cells would get into the central nervous system during disease and whether this would reduce that process. And you can see that nalfurafine targeted those T cells and reduced their ability, or either prevented them or got rid of them. We're not sure yet which. But reduced the presence of immune cells in the brain. When we looked at remyelination, one of the gold standards to look at those myelin sheaths is to use an electron microscope, because you can see, and if you look in the healthy, you can see the little diagram of the axon with the green myelin wrapped around it. And it looks remarkably similar to the other axons that are in that frame, that electron micrograph. Well, when you look at, in our MS model, you can see now that there are very few of those thick and luscious myelin sheaths. Now they're very small or they're absent. But when we treated therapeutically with nalfurafine and we activated that receptor, you can see now that those myelin sheaths are retained. And actually, we know that they had grown back because we looked earlier to show that at a peak disease, when there was destruction, that you would see that. And so this is all building it back from there, which was really, really very hopeful for us to look for something that can rescue that remyelination. Okay, I'll take the next one, please. So now we're at the stage where we're thinking, what do we do? Well, we have something we've repurposed medication, nalfurafine, which is incredibly safe. It is used, the other indication actually is for itch in hemodialysis, so it's a very different application. But we should go into a clinical trial. And so, for that we have designed a trial and we've got it approved, and now we're hoping to get that started as soon as we can get the funding and everything in place to do that. And really, for getting that money together and doing that, and the three of us have formed a company to help get that process going. So, what are we going to do? Well, we have to think about remyelination and how we're going to measure it. So as I say, it's always the difficulty in designing a trial is getting that primary outcome measure. So how do you measure remyelination? Well, it turns out, you know, when I talk about the central nervous system, you always think of the brain and the spinal cord, but the optic nerve is also part of the central nervous system. And in fact, that nerve is myelinated, and that nerve becomes demyelinated in some people during MS, causing optic neuritis, causing visual disturbances. So that gives us a little window into the brain. Okay, so we're thinking about, can we look into the optic nerve? Who do we recruit for this? And, well, I say who already. We have it actually already done. Now it's already, which is exciting to have it there. But then the other considerations really is, you know, this, as always, there's safety. We know a lot about the safety of this, which is helpful because there was that post-marketing survey that is often a phase 4 clinical trial. We know that's step 5, so we know a lot about the long-term effects with the treatment of this. So that's given us a lot of help. And so now it's about logistics. And so this project now is crucially at that point. And hopefully, we'll get it all together and be able to start. And I think the next slide should be acknowledgements. No, it's not. It is what have I learned. But fortunately, what I've learned is summarized really quickly. Could I have the next slide, please? It really is. Because if you think about it, developing that idea to a drug, it sounded really simple when I put 5 steps, you're right through. It's not quite as straightforward, and there are a lot of stumbling blocks. And a lot of those stumbling blocks, you can prevent some of those simply by better planning, by more thought, by taking your time. And I think that's one of the biggest things I learned is not to try to rush through it, to make sure each step is done properly. And repurposing drugs can assuredly accelerate the process. But we do have difficulty when we start thinking about protecting the ideas and developing it, but also the fact that it's not an invitation to jump into another indication. So always to move cautiously and to make sure that you have tested within the population that's relevant. And then I think we'll just go to the last one, which is what does it mean? What does it all mean? It means if you want to do it right, you take your time, you know, and we have to do it right. Okay. So I am going to just acknowledge quite a few people who have done most of the work. I've done very little of this work. I've talked about it a lot, but they've done all of this work, and they've been really brilliant. And I've been blessed to have them as colleagues, as students, and as staff. I'd also thank the funding bodies who have supported the research over the years. And you for sitting here as I blathered on, went through technical difficulties. Thank you very much.
I was wondering about, is that okay? I was wondering about the numbers that might be involved in the clinical trial process, particularly if you have to go through all 3 stages. What sort of gross numbers are we talking about? And where do you recruit them? And do you have a population in Wellington, or do you go wider through New Zealand or even outside of New Zealand? And are you allowed to reuse people? So you use them at one stage and then use the subject again later on?
Yeah, so the first trial for the clozapine and the risperidone was here in Wellington. And that was, ultimately, we would have recruited 36 people, okay, which, because we were looking for a very specific cohort, and so we were looking for people with progressive and a certain age, certain disability, you know, it's very difficult. And so for the trial for the remyelinating drugs, we would be looking at multiple sites, because that's the only way you can do it. Multiple sites in New Zealand and Australia, because of the numbers. And so MS has a very high frequency in New Zealand, but being a small country, it's still a very low number. Yeah, so it would be multiple trial sites doing that.
Can you reuse people?
How would I, reusing in what way? So
Use the same person at different stages of the same clinical process.
No, you would be only used once, because we need to see the effects in different people.
I mean, could you use in stage 3 someone who...
Oh, okay, I understand, doing different phases. I think it depends upon the trial and what the readout is. Okay.
Not quite. Yes, it does sound a bit low. I have a question too about the people you choose to use in your research. Almost every aspect of human physiology you come across, and particularly in terms of research, you find huge individual variation. That what works for one person, another person is 10 times the amount. Doesn't this bedevil your research hugely?
Mm-hmm. Yeah, so it is one of the things that you need to take account when you're doing clinical trials is that huge variability. And so when I started talking about sample size, you have to do a sample size calculation. You have to understand whether you have the power. And so when you go back to that primary outcome, what you measure as success, if it is going to be, say, remyelination, then you have to have a measure. Then you have to know how variable that is in the population, so that you can do a calculation, say, okay, well, I'd need 10 people to see a difference of this magnitude, or 100 or 1,000. You know, so that's why there's a lot of this is about the planning and trying to make sure that if there is a difference, you can detect it. Because you cannot run a clinical trial twice. You know, often you get the money for that trial, and if it fails for whatever reason, you are not going to get a second chance. It's very rare. And so you spend most of your time planning and making sure that if something's there, you can detect it. And variability is a huge problem. One way to do that is you can ask very specific questions in your clinical trial. So you can have a narrow definition, so you'd only have people that have this type of disease. You know, it limits your findings and how applicable it is to a whole population. But if there's something there, you may be able to find it. And that may give you the oomph to go to the next trial to understand and use a different population or a larger population. Yeah. But I think that's also why there's that phase 3, that post-marketing safety survey and monitoring. That happens because if you have a 1 in a million effect, you're not going to see that in any of these trials. It's not until it gets out there that you really start to understand what the effects are.
I was curious as to how the optic nerve is measured for progress with remyelination.
So there are a couple ways of measuring, but if you ever go to Specsavers and you look in there and they can look at the optic nerve and they can look at the nerve fiber, the layers. They can assess roughly myelination through measures there. But you can also look at, you can use clinical measures, because sometimes in a trial you want to know it has an effect, right? And just showing demyelination doesn't necessarily say that that's affecting your vision. So you might use something like visual acuity, how well you can see. And you can also measure the speed along the nerve axon. So you can measure electrical impulses from visual stimuli. And that's another way to give you like a functional, but again, the real important one is vision, right? But all of those can work.
Thanks for tonight. I just wondered, were you looking at people with progressive disease to get your cohort, or were you looking at people with similar symptoms?
So for the CRISP trial, it was looking in progressive disease. Yep. I'm not 100% sure what the criteria are for this, for the trial with the nalfurafine. I know it's a long list, and I don't know where they decided, because I'm not a neurologist, and it's gone to people whose expertise is in a clinical trial, because this would be a very large, multi-center one. My CRISP trial was a really small one. It was very lovely, but you give some things to the experts.
Does that allow you or nearly allow you to skip phase 1 because you've got the data from when it was used for another purpose?
Yeah, so yes and no. So when you think of the CRISP trial, it would have been considered, say, a 1B2A. So it was kind of between the two because it was about safety. And it may depend upon the medication. If the medication is used normally in a wider range of people that are very vulnerable, when you think of their physiology, potentially you can skip that. But you will often still build in a lot of safety aspects if you go into a new population.
My other question is, I know that doctors can prescribe for things that the drug is not approved for, and often do. Do you worry, or not you, but the clinical trials, worry about this, put in safeguards? What do you do to prevent a GP coming tonight and going, oh, I'm going to prescribe that tomorrow?
So I've had people say, oh, I've read that. How much do I do? You know, my GP will give this to me. Yeah. And I have to say that was probably the biggest learning for me from that CRISP trial with the clozapine, is that you can't just guess and say, okay, well, do this. We don't know the dose, and we don't know if there are going to be any effects. And we can't prevent. But I would hope that no GP would just randomly write out a prescription. And I don't think they would.
Okay. Thank you.
Thank you so much, Anne. That talk was incredibly informative, and it's just wonderful to hear how many people are working towards making things better with MS. It's an area I'm very passionate about. So thank you so much. What you're doing is amazing. And thank you to everybody here for coming along. It's wonderful to see you all, and to everyone who's joined the live stream. Thank you very much. Hopefully the technical difficulties didn't make too much of an impact. But yes, love to invite you to stay on for a cup of tea and some food, and thank you so much.